Today, we will be explaining about affinity maturation. Affinity maturation is when B cells increase the affinity for antigens, while maturation with the help of PFH cells in Japanese, ロホ性ヘルパー T 細胞. In this image, we see the parent cell on the left and the daughter cells on the right. When the B cells divide because of mutations, each daughter cells will have different antigen binding sites and therefore different affinity to a particular cell. As time passes, only the daughter B cells with the strongest affinity stays alive, and this is called affinity maturation. When there is antigen floating around, all B cells may respond. However, when there are less antigen to do the work of the immune system, only the daughter cells with the strongest affinity will live. We can say that affinity maturation is like natural selection of B cells that react to specific antigens. The stronger the affinity of the B cell, the more it survives. By affinity maturation, the cells can recycle inefficient B cells while holding on to the B cells with strongest affinity so that they can remove antigens more accurately and efficiently. Next, we'll explain the mechanism of affinity maturation. What becomes important here is somatic hypermutation. Somatic hypermutation occurs in the antibody gene. Some of the B region of B cells have mutations causing various affinity. More precisely, diminution of cytosine in DNA to uracil by an enzyme called AID causes DNA repair and the mutation happens. Only B cells with high affinity remain, and those are activated by TFH cells and turned into memory B cells or plasma cells. Germinal center is a temporarily formed structure within B cell zone in secondary lymphoid organs. First, activated B cells come into contact with FDC at the germinal center. Then B cells prey on the antigens on the surface of the FDC. After that, B cells present antigen to TFH cells, which are reactivated and proliferated. Since the number of TFH cells in the germinal center is limited, only highly competitive B cells can stably bind to TFH cells and receive T cell dependent survival signals. As we explained earlier, affinity maturation takes place within the germinal center, which is only alive temporarily. The process of germinal center formation begins with the antigen presenting cells such as dendritic cells and macrophages recognizing the antigen. After recognition, those cells move to the secondary lymphoid organs, for example, spleen, lymph nodes, tonsils, and present the antigen to T cells. At the same time, B cells are activated in the secondary lymphoid organs, recognizing the antigen and receiving help from helper T cells. Activated B cells move to the lymph fascicles and they're increased by T cell signals and form germinal centers. From what we've discovered so far, it is clear that affinity maturation mainly involves B cells. So without affinity maturation, the following effects can occur. First, a decrease in immune response can be expected because the antibodies are not optimized to target the invader. Second, immune memory will not function effectively. This also weakens the effectiveness of vaccines. As you can see, the lack of affinity maturation has a major impact on the immune system.